Welcome, dear students. I am Muhammad Rauf, lecturer, Department of Zoology Cast. Welcome to the course Principle in Animal Life 2. This is lecture number 17. The topic is sex-linked genes. Discussing agenda for the topic will be sex-linked genes exhibit unique patterns of inheritance, the chromosomal basis of sex, inheritance of X-linked genes. Examples. Lecture outcome. After watching and listening this lecture, students will know about sex-linked genes and its various aspects. Now, introduction. Sex-linked genes exhibit unique patterns of inheritance. Sex-linked genes are those genes which are located on sex chromosomes. For example, the sex chromosomes in the human female are XX and the human male are XY. So the genes which are located on these sex chromosomes are known as sex-linked genes. The identity of the sex chromosomes in an individual could be inferred by observing the sex of the fly. The behavior of two members of the pair of sex chromosome could be correlated with the behavior of the two alleles of the eye color. In our previous lecture, we discussed T.H. Morgan's experiments with Drosophila melanogaster. T.H. Morgan, for the first time, experimentally proved that genes are located on the chromosomes. In that case, he crossed red-eyed females with white-eyed mutant males, and at the end of the experiment, he concluded that the behavior of the genes which determine eye color is related with sex of the Drosophila melanogaster, means the offsprings having mutant eye color, that is white-eyed color, all of them were males and none of the females had white eye color. So he draw a conclusion that the genes which determine eye color are located on the sex chromosomes. So the sex chromosomes are the vehicles which provide a seat for the eye color genes in Drosophila, and if you analyze the behavior of sex chromosomes, you can attribute the same behavior to the eye color genes and their alleles because they are located on the sex chromosomes. Now, sex-linked genes exhibit unique patterns of inheritance. As you just learned, Morgan's discovery of a trait, white eyes, that correlated with sex of flies was a key episode in the Development of the chromosome theory of inheritance. Chromosome theory of inheritance was presented by Walter Sutton and Theodore Boveri. In that theory, they hypothesized that the Mendelian hereditary factors may be located on the chromosomes because the behavior of the chromosomes during meiosis is similar to the behavior of Mendelian factors. So they presented the hypothesis that the Mendelian factors may be located on chromosomes, and it was experimentally proved by T.H. Morgan that yes, the genes or the Mendelian hereditary factors do reside on chromosomes. Because the identity of the sex chromosomes in an individual could be inferred by observing sex of the fly, the behavior of the two members of the pair of sex chromosomes could be correlated with the behavior of two alleles of the eye color gene. In this section, we will take a closer look at the role of sex chromosomes in inheritance. The chromosomal basis of sex. Humans and other mammals have two types of sex chromosomes, designated as X and Y. The Y chromosome is much smaller than the X chromosome. A person who inherits two X chromosomes, one from each parent, usually develops as a female. We already know that the chromosome complement of the sex chromosome complement of a female is XX. So the female inherit one X chromosome from either parent. On the other hand, a male inherits one X chromosome and one Y chromosome. The male will always inherit the Y chromosome from the father and the X chromosome from the mother. Short segments at either end of the Y chromosomes are the only regions that are homologous with regions on the X chromosome. Now that's an important statement because during meiosis the homologous chromosomes align lengthwise gene to gene, and the synaptonemal complex is developed between them, and then crossing over takes place. So what about the sex chromosomes in the human male? We already studied here that the human male has one X and one Y chromosome, and the Y chromosome is much smaller than the X chromosome. So what will be the behavior of this pair, which is non-homologous, during meiosis, whether they will uh, show synapses and crossing over with each other or not. So this is the answer for you, that short segments at either end of Y chromosome are the only regions that are homologous with regions on Y chromosome. So although those these two chromosomes are different from each other, their gene complement is different from each other, but there are certain regions on X chromosome and Y chromosome which are homologous to each other, and these are the only points where the crossing over will take place between them during prophase 1 of meiosis. On the next slide, we observe the size of X chromosome, which is considerably greater than Y chromosome. And according to the data of the 2001 genome project, the human X chromosome carries more than 1000 genes, whereas the human Y chromosome carries very few genes, about 397, and the function of round about 100 of them is known. So have a look at the size of these two chromosomes. Y chromosome is much smaller than its X chromosome counterpart. So now we continue from the slide before that image. So there are certain regions on X chromosome which are homologous to certain regions on Y chromosomes, and these homologous regions forces the two chromosomes which are structurally different, whose gene complement is different, to behave like homologous chromosomes during meiosis in the testes. In mammalian testes and ovaries, the two sex chromosomes segregate during meiosis. You already know about this fact. Each egg receives one X chromosome. In contrast, a sperm fall into two categories, one X and one Y. So the human female is homogametic. The human female produces same type of gametes. All the gametes are X carrying, whereas the human male is heterogametic. The human male produces two types of sperm, an X carrying sperm and a Y carrying sperm. So remember, the human female is homogametic and the human male is heterogametic. We can trace the sex of each offspring to the events of conception. Now it's very easy to determine the sex of the individual which will develop from a zygote by knowing about the sex chromosome complement of the two gametes that fuse together. So if a sperm cell bearing an X chromosome fertilizes an egg, the zygote is XX, a female. If a sperm cell containing a Y chromosome fertilizes an egg, the zygote is XY male. Here the chromosome complement of only the sperm is shown. The egg is not shown because the sex chromosome complement of the egg is always X. So if X carrying sperm fertilizes the egg, the zygote is XX female. If Y carrying sperm fertilizes the egg, the zygote is XY male. So this is sex determination. Sex determination is usually decided at the time of fertilization, and there is another term which is called sex differentiation. Sex differentiation is the difference in the physical features of male and female, and when the zygote develops for a certain period, then it becomes, or it starts to develop features like a male or a female, depending upon what happened at the time of fertilization. But at the time of fertilization, whether an individual, whether a zygote will be a potential male or a potential female is 50-50, means there are 50% chances that a fertilized egg will develop into a male or a female. You can also say from that sentence you can conclude that in human male the X and Y chromosome carrying gametes are produced in equal number, and only then this 50-50 chance become possible. Now, in the last lecture we talked about Drosophila's sex chromosome complement. Drosophila has 8 chromosomes which are arranged into 4 pairs. 3 of them are autosomes, 1 of them is sex chromosomes, and the sex chromosome complement of Drosophila is same as that of the humans. The XY is male and the XX is female. Now there are different types of chromosomal systems, sex chromosomal systems, beside the XY which exist in Drosophila and humans. So let's have a look at other types of sex chromosomes systems. This is the humans, for the humans, and it is pretty much straightforward. 44 pairs of autosomes, 1 pair of sex chromosomes which is XY, heterogametic, 2 different types of gametes, human female, 44 autosomes, 1 pair of sex chromosomes. Sex chromosomes are of the same type, so they are homomorphic. Sex chromosomes are different from each other, they are heteromorphic. Human females is homogametic, all the eggs are X carrying. So then they have concluded that what will be the sex of the zygote, it depends which of the sperms fertilize the egg. Now this is the sex chromosome complement of grasshopper, cockroaches and some other insects. It is X naught. In the humans it is XY, in the grasshopper it is X naught. So what is meant by this naught? There is only one type of sex chromosomes. In the humans there are two types of sex chromosomes. In grasshopper only one type, and that is X. So then what will be the chromosome complement of male and female? So there is only one type of sex chromosome, the X. Females are XX, means female carry two X chromosomes as their sex chromosome complement. Male have only one sex chromosome, only one. There is no other sex chromosome. So the sex of the offspring is determined by whether the sperm cell contain an X chromosome or no X chromosome. This is very interesting. So here this is the sex chromosome complement of a male, means some of the sperms will be carrying X and some of the sperms will have no such chromosome, whereas the eggs produced by the female will carry X chromosome. So how the sex of the offspring will be decided? It depends that whether an X-carrying sperm fertilizes the X-carrying egg or a sperm with no sex chromosome fertilizes the egg. If a sperm with no sex chromosome fertilizes the egg, then obviously the zygote will have a single X chromosome and it will be a male. But if the X carrying sperm fertilizes the X carrying egg, then it will be an XX female. Now here is another sex determination system, which is ZW. In birds, some fishes and some insects, the sex chromosome present in the egg, not the sperm, determine the sex of the offspring. Now it is exactly opposite to the situation in the XY system. In the XY system, it is the sperm which determines the sex of the zygote, because the sperms are of two types. Here the sperm is of only one type, so all the sperms will be Z carrying, but there will be two types of eggs. So there is a heterogametic system in female and homogametic system in male, exactly opposite to the one we saw in humans and Drosophila. Here the egg will determine whether the zygote will be a male or a female. Now there is yet another system, the haplodiploid system. There are no sex chromosomes in most species of bees and ants. Females developed from fertilized eggs and are thus diploid. Males develop from unfertilized eggs and are haploid. They have no fathers. Now, the queen in honeybees has a special type of pouch where she stores sperms given by the drones, and it is decided by the queen whether to produce females or males. So when the females are intended, then the sperms are allowed to fertilize the egg, and when the males or the drones are intended, then the sperms are not allowed to fuse with the eggs, and the eggs develop on their own into an individual. So The drones in the honeybee are haploid, and here an interesting thing to share is that in drones the gametogenesis occurs through mitosis because the drones are already haploid, their somatic cells are haploid, their germ cells are haploid, so mitosis is enough to produce gametes and there is no need for further reducing the chromosome number. Now we discussed the sex determination which occurs at the time of fertilization. Now, how the sex differentiation occurs. The human male and female are physically different from each other. So, at which point during pregnancy the differentiation in the developing embryo starts, and it is 2 months old. When the embryo becomes 2 months old, then the sex differentiation starts. It starts to develop either features of male or female depending upon what was its chromosome complement during fertilization. So before then means before the 2 months of age of the developing fetus, the the rudiments of the gonads are generic, means they have the ability to develop both into testes or ovaries. These rudimentary gonads have the capability to develop either into testes or ovaries, but it is determined by the sex chromosome complement of the embryo. In 1900s, a British research team identified a gene on Y chromosome required for the development of testes. I hope you remember the name of that gene, which is SRY gene, sex determining region of Y chromosome. If SRY gene is absent, then these rudimentary gonads will not develop into testes and instead they will develop into ovaries. The biochemical, physiological, and anatomical features that distinguish male and female are complex, and many genes are involved in their development. In fact, SRY codes for a protein that regulate other genes. Regulate means that control the expression of other genes, whether other genes will express or not, if expressed, to which extent. Now researchers have sequenced the human Y chromosome and have identified 78 genes that code for about 25 proteins. Some genes are duplicates, means they are present in more than one copy. So here is yet another question, that how 78 genes code for only 25 proteins, because each gene codes for its own messenger RNA. So why not 78 genes code for 78 proteins? It is because of the structural complexity of the proteins. Some of the proteins are much complex and they are formed by combination of two different polypeptides. Remember the quaternary structure of proteins where two different polypeptides combine and form a protein. So about half of these genes are expressed only in the testes and some are required for normal testicular function and the production of normal sperm. In the next slide, we will analyze the events that results in the development of the fetus as a female or as a male. So this is an egg with X sex chromosome. If it is fertilized with an X carrying sperm, the result will be a female. If it is fertilized by a Y carrying sperm, the result will be a male. So let's suppose this X and Y fuse, it is XY. The genetic sex of the embryo is XY. Here the genetic sex of the embryo is XY. Now here the Y chromosome already discussed carry a gene SRY gene. SRY gene changes the undifferentiated gonads into testes, and if there is no XY chromosome present, that means there will be no SRY gene, so the undifferentiated gonads will automatically develop into ovaries, means the undifferentiated gonads by default develop into ovaries unless a Y chromosome is there. So once the testes are produced, the testes secrete masculinizing hormones. Masculinizing means which brings about male uh, primary sexual characteristics like testosterone. Here, ovaries does not produce androgens. So, in the presence of testicular hormones, means in the presence of testosterone, the undifferentiated reproductive tract and external genitalia develop along male lines, means like the ones which are there in the males. With no masculinizing hormone, undifferentiated reproductive tract and external genitalia develops along female lines, means like the ones in the females. So a gene located on either sex chromosome is called a sex-linked gene. Now sex chromosomes are of two types, X and Y, so the genes located on Y chromosomes will be Y-linked and those located on the X chromosomes will be X-linked. The Y chromosome is passed along virtually intact from a father to all his sons, means there is no change in the gene complement of the Y chromosomes, and exactly the same Y chromosome is passed to the offsprings. So we can deduce that although the Y chromosome has certain homologous regions with the X chromosomes, which enables them to pair up during meiosis and exchange segments. So these segments are not of any significance. Because there are so few Y-linked genes, very few disorders are transferred from father to son on the Y chromosome. Now, this is an interesting sentence which says that the Y chromosome does not carry too many genes. It only carries 397 genes, and the function of round about 100 of them is known, and majority of those genes are uh, responsible for determining the maleness and the fertility, and no such genes, disease related genes, are located on Y chromosome. So the father does not transfer the disease related Y linked genes to their sons. A rare example is that in the absence of certain Y-linked genes, an XY individual is male but does not produce normal sperm, because already discussed Y-linked genes are responsible for determining fertility and maleness. So if problem occurs in those genes, the only consequences is its effect on the fertility of the individual. Human X chromosome contains approximately 1100 genes which are X-linked. Is the human X chromosome carry 1100 genes. The fact that males and females inherit a different number of X chromosomes leads to a pattern of inheritance different from that produced by genes located on autosomes. It means that the sex chromosome complement of X and Y is different. So the male and female are vulnerable to different extents for any defects in the those sex-linked genes, whereas the the gene complement on autosome is same. So the only difference is the gene complement of the sex chromosomes and the sex chromosome combination in both sexes. So the pattern of inheritance is different, which we will study in the coming slides. Now, inheritance of X-linked genes. While most Y-linked genes help determine sex, the X chromosomes have genes for many characters unrelated to sex, means the genes which are located on the X chromosome have functions other than just controlling the sex. X-linked genes in humans follow the same pattern of inheritance that Morgan observed for eye color locus in Drosophila, means females have two X chromosomes, male have only one X chromosome. So the terminology of dominant and recessive for X-linked genes exist only in human females. In human males, no such terminology exists for X-linked genes. And the human male is hemizygous for X-linked genes. where the X-linked genes in the human male does not have another partner. We say that the genes exist in the form of alleles. So this statement does not apply to the X-linked genes in the human male. They are only present in the single copy. Father pass X-linked alleles to all of their daughters, but to none of their sons. Sons or a male's sex chromosome complement is XY, so the father will always donate the Y chromosome to the sons, and the father will never donate X chromosome to sons, whereas the father will always donate its X chromosome to daughters. In contrast, the mother passes its X chromosome both to its sons and daughters for the inheritance of a mild X-linked disorder, red-green color blindness. Now, if an X-linked trait is due to a recessive allele, a female will express the phenotype only if she is homozygous for that allele. Means recessive alleles are the ones which only exhibit their effect if two copies are present. So, suppose there is a disorder which is X-linked, means the gene of that disease is located on X chromosome. So, and this disorder is recessive. So, females will be carriers for this disorder because the females have two X chromosomes. So, one X chromosome will carry a normal copy, the other will carry a defective copy. The female will be healthy themselves, but they will be a carrier. On the other hand, the male only has one X chromosome. So if the allele located on that single X chromosome is defective, the male will show symptoms of that condition. So as already stated, that the terminology of dominant and recessive for X-linked genes only exist in human females. In the human male, there is only one X chromosome. So dominant and recessive disorders both will be equally expressed. Now, any male receiving the recessive allele from his mother will express the trait. Mother, if a mother is a carrier, means allele on one of the X chromosomes is defective and allele on the other X chromosome is normal. So if the son receives that X chromosome which is carrying a defective allele, then the son will exhibit the symptoms of the disease. And for this reason, far more males than females have X-linked recessive disorders. This is also a good statement. For a recessive X-linked trait to be expressed, both the copies should be defective, that is in females. But a male only, because in male only one copy is present, so if it is defective, the male will show the symptoms of the condition. So X-linked disorders are much more common in males than in females. A number of human X-linked disorders are much more serious than color blindness, such as Duchenne muscular dystrophy, which affects 1 out of 3,500 males born in the US. And the disease is characterized by a progressive weakening of muscles and loss of coordination. Affected individuals rarely live past their early 20s. Now, researchers have traced the disorder to the absence of a key muscle protein called dystrophin and have mapped the gene for this protein to a specific locus on X chromosome. Hemophilia is another disorder which is X-linked and recessive, means more males will be vulnerable to acquiring this disorder than females. And hemophilia is also known as bleeder's disease because the blood clotting factor is missing in those individuals and they can bleed to death from a minor injury. And once again, we come across the intermarriages. We already discussed that during our lectures on meiosis, where the consanguineous marriages quite often results in offsprings which carry genetic disorders because quite often the families have a defective copy and the members of the family are themselves healthy and carriers, but their offspring receive defective copies from both parents and develop the disorder. Now, here is the inheritance of X-linked genes in the female. Both the genes for color blindness, there is no defective genes, means the female is normal without carrying a defective copy, but the male is carrying a defective copy. Then we perform a cross, so the outcome of the cross is the females will be carriers, whereas the male, if receive the normal copy, then they will be normal. And in that case, if the mother has both normal copies, then it will not affect the males because the father only transfer Y chromosome to the sons. Now in other examples, now in this example, the female is a carrier. In the previous example, the female was healthy without any defective allele for the color blindness. Here, the female is carrier and the male is normal. So what will happen to their offsprings? 50% of the offsprings will be normal, 25% will be carriers, and all the sons will be, and 25% sons will be having this condition. So there is a 50% chance that each daughter will be carrier like her mother and 50% chance that each son will have the disorder. 50% chance normal carrier, 50% chance normal diseased. Now here is yet another example and probably a consanguineous marriage where both the female is carrying a defective copy and the male is already affected. So what will be the consequences if the couple bears a child? There is a 50% chance that each child born to them will have the disorder regardless of sex. Means maybe male, maybe female daughters who have normal color vision will be carriers, whereas males who have normal color vision will be free of the recessive allele. That's the end of the lecture. Thank you very much. Allah Hafiz.